confirmation that I'm connected. If you can see me live, okay. So this session, this series of session, as you know, is AIIMS marathon, and we will be discussing various AIIMS question of past 7-8 years. So without ado, we will just start with the questions. So what will happen is I'll show you a question and you will have to answer it, and maybe we'll discuss the options and some amount of theory with each of the questions. Okay, so let's get started. Now this session is sponsored by Unacademy, NEET PG Plus. Okay, and we'll talk about what is Unacademy and how it can help you in your NEET PG preparation. But before that, let's start with the question. This is the first question. Nitrogen narcosis is caused due to. So if you know the correct answer, write in the comment below. Right? Nitrogen narcosis is caused due to. A is nitrogen inhibiting dismutase enzyme. B is increased production of nitric oxide. C is increased solubility of nitrogen in nerve cell membrane, and D is decrease in oxygen free radicals. If you know the answer, write in the comment below. Very good, Dr. Dilpreet, very good, Dhananjay. So the correct answer here is increased in the solubility of nitrogen in the nerve cell membrane. So what happens is, when you go deeper into the water body, I mean, suppose if this is the sea level, as you go deeper, because the pressure of the water, so the nitrogen becomes more soluble, and it is dissolving in the nerve cell membrane, which will cause all this symptom. Okay, so let's look at this of the following. Which of the following is not degraded by colonic flora? So options are pectin, lignin, starch, and glucose. Which of the following are? So Dinesh says C, Dilpreet says B. Anyone else? Matt for AIIMS says B. Manik says C. Okay, so there seems to be a confusion between starch and starch and lignin, but most of you have answered this correctly. The correct answer is actually lignin. Lignin is very different than all the other. These are all dietary fibers basically, starch, pectin, and but lignin is very different. It is neither digested nor degraded. Okay, so that is lignin. So the correct answer here is lignin. Okay, starch can be degraded by colonic bacteria. Let's look at this question. Lac operon. This question is around lac operon. CAP, that is C, catalytic activated protein in lac operon, is a positive regulator, negative regulator, attenuator, or in under constitutive expression. So one last chance says B. Dhananjay says B. Shatish says A. Anyone else? Anyone else? So the correct answer here is A. The correct answer here is A, positive regulator. C, catalytic activated protein act as a positive regulator. So only Satish has got it correct. Rest all are thinking as a negative regulator. So what happens, I'll tell you in short, because most of you got it correct. So what happens is, whenever this is related to the so what happens is, whenever the level of glucose falls down, the level of cyclic AMP increases. The glucose level and cyclic AMP level are inversely proportional. So as the level of glucose is falling down, the level of cyclic AMP actually increases, and this cyclic AMP with catalytic activated protein actually goes and attaches to the promoter region and even activates it. Okay, so it is acting as a positive regulator. Okay, so remember, it is a positive regulator. Let's answer this question. ApoB48 and ApoB100 is synthesized from the same RNA. The difference between them is due to. This is again a very important question. Try and answer this question. So Meena says A. Anyone else? Dilpreet also says A. RNA splicing. Matt for AIIMS says A. One last chance says A. Dinesh says A. Very good. So RNA splicing and RNA editing are two different things. Okay. Anyone else? B. Allelic exclusion. No. Still the correct answer I have not got. No. Zahedi, it's wrong. It's not A. Very good, Yagu. Yagavan Yagu got it correct. Very good. The correct answer is basically deamination of cysteine to uridine. This is an example of basically RNA editing. Okay, RNA editing. So this is a post-transcription modification, and this so I am not going to go very detailed into the description of it. So I'll just explain you a little. So what happens is RNA editing is an exception to the our central dogma. What happens is normally if you see there is never a nitrogenous base change at RNA level, is never happening. But in RNA, so nitrogenous base can be removed, okay, but it is never changed. Okay, so splicing is just removing. Okay, but in RNA editing actually there is a change at nitrogenous base. So what happens, a correct example is apolipoprotein B. So apolipoprotein B has 4564 codons, and the codon of interest is 2153. That codon is CAA. Okay, now what happens, based on different organs, it is edited differently. So in liver we have got the unedited mRNA, and which will give to the complete protein, that is apolipoprotein B100. Now this apolipoprotein B100 we know transfers triglyceride from liver in throughout the body. Okay, and it will consist of all the amino acid, that is 4563 amino acid. But in intestine what happened, that CAA, which gives to glutamine, is deaminated by cysteine deaminase, and this CAA is converted into UAA, which is a stop codon. Yes, Dr. Pranab Paul. So this stop codon actually stops the protein polypeptide at 2153 codon level. So we have only 2152 amino acid, which is 242 kilodalton, and it is apolipoprotein B or apoprotein B48, and it is basically a part of chylomicron. Okay, so this is RNA editing. It is very different from splicing. Okay, in splicing the change in nitrogenous base does not happen. Here the nitrogenous base itself is changed. Is everybody clear about RNA editing? Is everybody clear about RNA editing? Okay, good. So let's, other examples of RNA editing are glutamate receptor, where glutamine is changed to arginine, and triposome mitochondrial DNA. Let's look at this question. All of the following are hydrophilic hormones that act on cytosolic receptor except one, which is lipophilic, and act on nuclear receptor. So you have to tell me. Good, Devashri. Thyroxine. Matt for AIIMS says thyroxine. Anyone else? Good. Good, Dilpreet. So I think all of you have got this correct. Okay, all of you have got this correct. So it is the thyroxine which is which acts on nuclear receptor. Rest all are acting on cytosolic receptors. Very good. This question all of you have correct. Now this again is a genetic question. Try and answer this question. Histone acetylation causes. I hope the pace is okay. I'm not spending a lot of time on each question. I want to discuss maximum question. Okay. So if you want me to explain any question, histone acetylation cause increased heterochromatin formation. No, Matt for AIIMS, it is wrong. It is not A. Anyone else? Dilpreet also says A. Anyone else? Good, good, good. Matt for AIMS. This is the correct answer. D is the correct answer actually. Okay, because what happens? Acetylation will be activating it, and methylation will be deactivating or silencing a gene. Remember this important concept. Okay, histone acetylation will be causing activation. So activation of genes. So euchromatic formation, and if it is methylation, then it will be leading to gene silencing. Okay. So basically this question is around epigenetics. So epigenetics are reversible, heritable chemical modifications of DNA or histone or non-histone proteins that do not alter the DNA sequence itself. Now it can be either a post-translational modification of histone, and a wide array of post-transcriptional modifications is there, which is called as histone code. Remember, these are dynamic and reversible, and they are heritable. So all these are very, very important. They are passed from generation. I mean, they are heritable, but they are reversible. Okay. Example is acetylation, deacetylation, methylation, phosphorylation, and ADP ribosylation. And then we have got DNA methylation, where a methyl transferase enzyme will methylate it, and which will lead to gene silencing. Okay. Let's answer this question. Triple helix DNA is due to. Very, very frequently repeated question in multiple PG entrance exam. It has come. Triple X DNA is due to Hochstein pairing, palindromic sequence, large number of guanosine repeats, or polyprepamide track. Anyone? So Debashri Panda says A. Anyone else? Okay. Satish says D. So the correct answer is actually A. Okay, correct answer is A, Hochstein pairing. Okay, so in this what happens, it is basically seen in RNA only, and what happens is in DNA, what happens is the nitrogenous base pairing is the same as the normal. So A will be paired with T and C will be paired with G, but there is an abnormal pairing. Okay, so because of which it is able to pair with two strands. The correct answer is A. Hochstein was the scientist who actually discovered it. So it's called as Hochstein pairing. Let's look at this question. Acetyl-CoA can be directly converted to all except. Try and answer this question. Acetyl-CoA can be directly converted to all except. Okay, Ijja says glucose. Anyone else? Satish says cholesterol. Satish, cholesterol, the first step itself is acetyl-CoA plus acetyl-CoA. So cholesterol can easily be formed from acetyl-CoA. Okay. So all these three, okay, fatty acid can be formed. Cholesterol, ketone bodies. Glucose, it can never form. What happens? Acetyl-CoA never takes part in gluconeogenesis directly. In gluconeogenesis, it never takes part directly. Okay, but how will it help in gluconeogenesis? It affects gluconeogenesis in two ways. First of all, it activates the first enzyme of gluconeogenesis, that is pyruvate carboxylase. Okay, so that it will activate, and second, it will go in under Krebs cycle to produce a lot of energy. We know that this energy, very good Dilpreet. So what happens? It goes in Krebs cycle and produces a lot of energy. This acetyl-CoA will go into Krebs cycle and produce a lot of energy, which is again helpful in gluconeogenesis. So that is how it will be helping in creation of glucose, but it never be directly. The question here is directly converted into. So it was never directly converted to glucose. Okay. No, it is not about changing into oxaloacetate first, Dilpreet. Remember, no way it is. This, when it goes into Krebs cycle, what happens? Both the carbons are actually broken down and released. So no way it is actually contributing to the carbon skeleton of gluconeogenesis. Remember this. Okay. Let's look at this question. After digestion by restriction endonuclease, DNA strand can be joined. So which enzyme is actually joining DNA strands? Okay, good, Ijja. The answer you are saying is B. Anyone else? Ligase, good Matt for AIIMS. Mehboob, good. Dilpreet, good. Meena, good. Good Darshini, good Dhananjay. Very good Nilima. Okay, great guys. I mean, all of you have got it correct. Okay, so the correct answer here is DNA ligase. Okay, right. So can anyone tell me what is the function of DNA polymerase? Quickly. DNA polymerase, what does it do? So in genetics, DNA replication, very good. The most common questions are asked around the roles of various enzymes. So first we have got topoisomerase. It relieves the torsional strain. So when helicases does the unwinding of DNA, does the unwinding of DNA, what will happen? There will be a torsional strain. So that is removed by topoisomerase. Okay, helicases will be ATP driven unwinding of DNA. Single strand binding. Okay, again it will help in unwinding of DNA. DNA primase will initiate the synthesis of RNA primers. DNA polymerase will help in DNA polymerization. Okay, and DNA ligase will seal single stranded nick on lagging strand. Okay. So this is a little tricky question. It's a favorite AIIMS topic also to ask. So let's answer this question. True about sickle cell disease are all except. True about sickle cell disease are all except. So single nucleotide changes result in the change of glutamine to valine. Sticky patch is generated as a result of replacement of non-polar residue with polar residue. HBS confers resistance against malaria in heterozygous, and RFLP results from a single base change. Good. Brilliant. Every one of you have got it correct. Very good. The correct answer is actually B. Right? The correct answer is B. Now if you see the options either B or A, any one of them has to be false, because here it is saying glutamine to valine. We know this is polar and this is non-polar or hydrophobic. Okay, and here it is saying non-polar is changed with polar. Okay, so actually this is correct. This is wrong. Polar is changed with non-polar. So if you see the molecular defect of sickle cell anemia, so this is the normal GAG and CTC, which is converted. This A is converted into T. So now the pairing is GTG and CAG. So we will have the RNA of GAG and GUG in case of sickle cell anemia, which will lead to the protein glutamine and valine. Okay. Now if we see this RBC, when there is deoxygenation under low oxygen tension, it will become what you call sickle cell, and it will undergo hemolysis. Very good. Let's answer this question. Gluconeogenesis in fasting state is stimulated by. Gluconeogenesis in fasting state is stimulated by. Anyone else? So there seems to be a confusion between D and B. So let me just explain. So first of all, we remember that whenever there is a fasting state, what will happen? Triglyceride, this is our triglyceride molecule. Okay, this will be broken down, and we will have these fatty acids which will undergo beta oxidation to form acetyl CoA. Okay, so whenever there is high acetyl CoA in the system, which means fatty acid breakdown has happened. And when will fatty acid breakdown happen? When the glucose level is falling down. Okay, so high level acetyl CoA indicates low level of glucose indirectly for the body. So this high level acetyl CoA will activate the first enzyme of gluconeogenesis, that is pyruvate carboxylase. Okay, so this is the correct answer, right? Now, pyruvate kinase is stimulation by citrate. Okay, this is not correct. And even this, pyruvate kinase by fructose 1,6-phosphate. All these are wrong. The correct answer is B. Very good. Let's look at this question again. In human, the genetic code is represented by a code of 3 nucleotide. If one amino acid is more than one triplet, this is known as. It's not ambiguity. Remember, the genetic code is not non-ambiguous. Okay, it is called as degeneracy. Degeneracy, not ambiguity. Degeneracy is when more than one triplet can code for one amino acid. So simply imagine one amino acid can be coded by more than one genetic code or triplet. More than one triplet can code for one amino acid. So this is called as degeneracy. Another thing is this triplet, this triplet will code only for this amino acid. So one triplet will code for only a specific amino acid. So our genetic code is not ambiguous, it is non-ambiguous. Non-ambiguous, because one specific triplet will code for that specific amino acid. Okay, very good, very good. So let's look at this question. Which of the following is true regarding fluorescence? Which of the following is true regarding fluorescence? Okay, I think there is an issue here. Both B and C are actually same. Okay, so sorry for the typo. Okay, so this is correct. Both B and C are correct. Okay, release of longer wavelength of light on absorbing shorter wavelength of light. Both are same and both are correct. Okay, so there was a typo. Let's answer this question. Ketone bodies are not used by. Ketone bodies are not used by. Quickly. Very good, very good. So all of you are very clear, and because RBC does not have mitochondria, so ketone bodies are not used by. Can anyone name another organ which does not use ketone bodies? Another very important organ which will not use ketone bodies. Quickly. Another very important organ. Is it liver or is it kidney? Brain is already there. Heart generally prefers ketone bodies. It is liver. Liver also does not. Liver is involved in production, but it never uses ketone bodies. Okay. Let's look at this question. Which of the following is known as a suicidal enzyme? Very good. Which of the following is known as a suicidal enzyme? Anyone else? Matt for AIIMS says B. Anyone else? Dhananjay also says, Devashree also says B. Anyone else? Mehboob says A. Anyone else? Okay, the correct answer is correct. Very good. The correct answer is actually B. Cyclooxygenase. So what happens? The product only destroys the enzyme. Okay, so this is called a suicidal enzyme. Let's look at this. Carbohydrate metabolism. Irreversible steps of glycolysis are. Very good, Nabda. Very good, Meena. Very good. So the correct answer is all of the above. Very good. So I'm not going into the entire discussion. Let's look at this question. This is the last question for today. A patient presents with von Gog's disease and ketosis was detected on investigation. all of the filings would be associated except. So remember the question is except. Okay, you have to find the except. So which of the following is associated except? So we have to find which of the following is false. Okay. So what is the defect in von Gerg's disease? Sorry, I have been allotted biochemistry, Darshini. So von Gerg's disease. What is the basic deficiency? The glucose 6-phosphate is not converted into glucose. So this is the issue. So what is happening? This enzyme, okay, this enzyme is basically inhibited. Glucose 6-phosphatase is inhibited. So what will happen? There will be a huge increase in glucose. There will be a glucose 6-phosphate, and what it will do? It will activate glycogen synthase and inhibit what? Inhibit glycogen phosphorylase. So there will be increased, glycogen synthesis and decreased breakdown, which will lead to hepatosplenomegaly. Plus, this step is also the last step for gluconeogenesis. So gluconeogenesis will also be impaired. So this also will be seen. Okay, this is true. This is true. Because so there will be severe hypoglycemia. Remember, von Gerg's disease, very, very severe hypoglycemia is seen. So this is also true. Last is low fat metabolism. No, the fat metabolism will be actually high, because what is happening? The glucose level is falling down. So what it will do? Glucagon will stimulate, and then there will be a lot of mobilization of fat. Okay, the fat mobilization will actually increase. So there will be hyperlipidemia. Okay, can anyone tell me why lactic acidosis is also seen? Why lactic acidosis is seen in cases of von Gerg's disease? Yes, very good. Yeah. Why lactic acidosis is seen in von Gerg's patient? Remember, this gluconeogenesis is inhibited, so this pyruvate is not being converted into glucose. So lactate also will not be able to convert it into pyruvate. Okay, this entire metabolism will be stopped. So they will be increased in lactate level, and so lactic acidosis will also be seen. So this is about von Gerg's disease. Okay, so all these I have explained to you. Okay, because what is happening is glucose 6-phosphate to glucose is being so increased glycogen production, reduced glycogen breakdown, progressive hepatomegaly, severe hypoglycemia, renomegaly, hyperlipidemia as fat metabolism increased, lactic acidosis as lactic does not get converted into glucose, presents at 3 to 4 months. Can anyone tell me the glycogen which is deposited in von Gerg's disease? Is it normal glycogen or abnormal glycogen? Is it normal glycogen or abnormal glycogen in von Gerg's disease? Very good, very good. No, it is normal. It is normal. Remember, only the glycogen phosphorus is just activating glycogen synthase and inhibiting this. Okay, all the glycogen formation and breakdown enzymes are working normally. So there will be increased amount of normal glycogen. Okay, this is very important concept. Only when there is a problem in debranching enzyme or branching enzyme, we will have abnormal glucose. Remember this very important concept. Only when there is a branching enzyme problem, then only we will have abnormal glucose. Okay, otherwise we will have normal glucose. Okay, so this brings us to the end of this session. We have been allotted 30 minutes. So hope you really liked this session. Okay, and in case any one of you is planning to join Unacademy, AIIMS batch. So there is an AIIMS specific batch which is going to be started. Okay, so if you're planning to join that, then you can use this code. Okay, so hope all of you know about the Unacademy learning app. So you can use this code. Okay, you can join a 3-month program before AIIMS, because a lot of AIIMS based discussion is conducted by all the faculty, all the top faculty is conducting AIIMS based classes where in detail. Now here I am just discussing question answer, there I will be taking you deeper into the concepts so that all the high yield topics of AIIMS are deeply covered. Okay, so if you are interested, here is a code which you can use which will give you a 10% discount. Okay, and moreover, if you use this code, Dr. Abhishek Gupta, just let me know. I will give you an access to my AIIMS PG exam paper as a you know as a gift from me. So we have an AIIMS PG exam question paper app where all the 10 years of all the AIIMS paper are there, all the even including the latest paper that I'll give if anyone uses this code Dr. Abhishek 10 YT or Dr. Abhishek 10. Okay, and